A man hears a joke, laughs, and suddenly his knees buckle, his head drops, and he collapses to the floor fully conscious, fully aware of everything happening around him, but completely unable to move a single muscle for several seconds. This is cataplexy, one of the defining and strangest symptoms of narcolepsy, a neurological sleep disorder in which strong emotion can trigger sudden, total loss of voluntary muscle control, and in which the boundary between sleep and wakefulness becomes so unstable that patients can fall asleep instantly, at any moment, with essentially no warning at all. Narcolepsy is fundamentally a disorder of sleep-wake state regulation. In a healthy brain, the transitions between full wakefulness, non-REM sleep, and REM sleep, the stage most associated with dreaming and characterized by the muscle paralysis called REM atonia, discussed at length in research on sleep paralysis, are tightly and reliably coordinated by specific brain circuits, ensuring that these distinct states occur in their proper sequence and remain stable once entered. In narcolepsy, this coordination breaks down. Patients can transition directly into REM sleep, or experience intrusions of REM sleep characteristics, including its associated muscle atonia, while still behaviorally awake, producing both the sudden, irresistible sleep attacks characteristic of the disorder and, in a significant proportion of patients, episodes of cataplexy, in which fragments of REM sleep muscle paralysis intrude directly into waking consciousness, typically triggered by strong emotional experiences such as laughter, surprise, or excitement. The breakthrough in understanding narcolepsy's underlying cause came through research converging from two initially separate lines of investigation in the late 1990s. Researchers studying a rare strain of narcoleptic dogs, which display symptoms strikingly similar to human narcolepsy including cataplexy-like collapses, identified a genetic mutation affecting a specific neurotransmitter system involving a molecule called hypocretin, also known as orexin. Around the same time, separate research on genetically engineered mice lacking the ability to produce orexin found that these mice displayed narcolepsy-like symptoms as well, providing strong converging evidence implicating this specific neurotransmitter system and the disorder. Orexin, produced by a small, specific population of neurons located in the hypothalamus, plays a central role in promoting and stabilizing wakefulness, and in maintaining the normal, stable boundaries between sleep states. Subsequent research examining the brains of deceased human narcolepsy patients, led significantly by researcher Emmanuel Mignot and colleagues, found a striking and consistent finding. Patients with narcolepsy accompanied by cataplexy showed a dramatic loss of these orexin-producing neurons, in many cases, a loss of 90% or more of the normal population of these cells, compared to the brains of people without narcolepsy. Without an adequate population of orexin-producing neurons to stabilize wakefulness and properly regulate the boundaries between sleep states, the brain's sleep-wake switching mechanism becomes fundamentally unstable, prone to inappropriate and sudden transitions into sleep-related states, including the muscle paralysis of REM sleep intruding directly into waking life as cataplexy. The leading and now well-supported explanation for why these specific orexin-producing neurons are lost centers on an autoimmune mechanism. Substantial evidence, including a strong and well-documented genetic association between narcolepsy and specific variants of immune system genes called HLA genes, points toward the body's own immune system mistakenly identifying and destroying its own orexin-producing neurons, in a manner conceptually similar to other recognized autoimmune diseases in which the immune system attacks a specific, normally protected population of the body's own cells. This autoimmune hypothesis gained particularly strong and unfortunate real-world support following an unusual cluster of narcolepsy cases that emerged in several European countries, particularly Finland and Sweden, following vaccination campaigns using a specific influenza vaccine called Pandemrix during the 2009 H1N1 influenza pandemic. Subsequent epidemiological and immunological research found a significantly elevated risk of subsequently developing narcolepsy among vaccinated individuals, particularly children and adolescents, with later laboratory research identifying specific molecular similarities between certain components of the vaccine and the orexin